Before I begin, I would like to give you a glimpse of my scientific journey. I obtained my bachelor's and master's in integrated MSc from Indian Institute of Technology, Kharagpur. During my degree, I had an opportunity to spend my summers working as a research intern in academic and industry labs across the world. My expertise is in self-assembly, and I'm currently specializing in self-assembled systems. In my research at UVic, I apply my prior knowledge in photochemistry to study and understand self-assembled systems at the fundamental level. I'm happy to be here today and to share some of my expertise that I gathered in this arena. Sorry. The agenda for today is I will cover some of the introductory concepts in self-assembly, and I'll talk about current applications, challenges in the drug delivery field, and how I'm addressing some of these challenges using light as a tool. Here is a picture to show you the length scale where chemistry and biology happens. The chemists deal with molecules typically in the range of 0.1 to 1 nanometers, as you can see in the figure. And they're pioneered in making molecules using various methods, be it from natural products, chemical synthesis, or using metals. As you see, biology happens at a different length scale far from the chemical scale. There has been a huge interest garnered in the recent years to make molecules around 10 to 100 nanometers, and these molecules will be referred as nanostructures in the rest of my talk. One example where the nanometer length scale becomes prevalent is in cancer research. Many of us in the audience might have heard about EPR effect. For those who doesn't know, this is a passive approach where drugs get delivered in the cancer cells by avoiding normal cells. Drugs we have are in the typically of the size of 1 nanometers. At this size, the drugs can enter normal tissue as well as a tumor tissue, as you can see in the figure. Reports show that the cancer cell has a leaky vasculature. As you can see here, the gaps shows us that the vasculature is leaky. And one way we can selectively deliver drugs to the cancer cell by avoiding a normal cell is by loading drugs into carriers, typically of the size of 10 to 100 nanometers. This is a size that can only allow them to enter the tumor cell and avoid a normal cell. As you can see in the figure, the bigger nanoparticle here couldn't enter the normal cell, whereas it can enter the tumor cell through the leaky vasculature of the blood vessel. However, making these nanostructures is hard because we are no longer dealing with single molecules. We are dealing with a cluster of molecules which resulted in this nanostructure. As we move up the scale in the length, complexity situation arises, and this complexity provides a chance to make more mistakes. Therefore, to build nanostructures, we need a process that's more efficient than current existing methods in the synthetic labs. Looks like biology has a built-in mechanism to build complex structures. Let's take an example of protein folding. Protein folding occurs at a rate of 10,000 nanoseconds per protein with 2 million types in humans. Biological processes such as protein folding and DNA replication are extremely efficient, far more complex than man-made objects, and hardly ever make mistakes. These life processes taught chemists to stop synthesizing everything from the scratch, rather that molecules can form nanostructures by themselves when provided with right conditions. In scientific world, this process is known as self-assembly. I would like to quickly tell you an example of self-assembly we see in our day-to-day life. Soap is made up of a molecule known as sodium dodecyl sulfate. It's a surfactant. Soap by itself doesn't have any cleaning action. When we dissolve soap in water, the SDS molecules in the soap aggregate and form a nanostructure in the form of micelle, and this micelle is responsible for the cleaning action of the soap. Unlike molecules formed by chemical bonds, the structures formed by self-assembly are held together by weak physical interactions. Because the molecules are held together by weak physical interactions, they tend to be reversible. The reversible nature of self-assembly allows components to adjust their position once they are formed, leading to more efficient and error-free process. This is exactly the process that we are looking for to build nanostructures. Self-assembly played a prominent role in the development of nanotechnology and expansion of microtechnology. Key applications of self-assembled structures are found in energy transport, as you can see in the figure over here, and also in microelectronics for building of semiconductors and electrode and devices. Also, the medical applications such as drug screening, diagnosis, imaging, and drug delivery. In this talk, I'm going to concentrate more on the drug delivery side of things as it is related to my research area. The molecules that are shown in the left here are drugs, our anti-cancer drugs that are in the clinic, like paclitaxel, fluorouracil, and camptothecin. In traditional drug discovery process, also known as structure-aided drug design, scientists make use of structure-function relationships to be able to design the drugs. For example, in areas such as computational chemistry, biological targets are defined first, followed by the identification of active sites that are responsible for the function. Once these active sites are identified, scientists design molecules that actually fits into these target sites through docking studies. In most cases, drugs we deal with these days no longer looks like this, but rather they look like this, in which a drug is present inside a nanostructure. Here, I show you the picture of a formulation which is approved for cancer therapy, known as the Doxil. It is made up of, the Doxil contains the doxorubicin, which is an anti-cancer drug enclosed in a liposome made up of MPEG-DSPE. This nanostructure typically is in the order of 100 nanometers. As we begin to move from molecules to nanostructures, the structure-aided drug design is no longer valid. Here is an example of self-assembly process indicated by using blue and yellow building blocks. The systems you see here in the made by self-assembly are not made from fixed parts, and the structure, as you can see in the figure, is changing and persistent only for a certain time. Because self-assembled systems are constantly moving, structure is not the sole contributor to the function. Understanding dynamics is a critical piece in self-assembly because the interactions of building blocks produce new functions that cannot be determined solely by studying the structure. Based on my experience as a bench chemist, seeing both traditional and latest drug development processes, I couldn't stop emphasizing the importance of studying dynamics can have in the way we develop self-assembled systems. For example, how the mobility of drugs is affected by the changes in the dynamics could give us crucial information about how fast or how slow the drugs can be released from the structure. By now, I think I convinced you enough that structure and dynamics are key to understand function in self-assembled systems. In my research, I use light as a tool to understand structure, dynamics, and function in the self-assembled systems. The figure here is a figure from known as Jablonski diagram, and I'm going to quickly take you through what happens when a molecule absorbs light. So when a molecule absorbs light, it goes from the ground state, which is S0 indicated in the figure, to higher energy level, which is S1. The molecule spends some time in S1. Typically, it's in the order of 10 to 100 nanoseconds and returns to the ground state. During this process, it emits either light or heat or both. Solid line from S1 to S0 indicates the processes which emits light, and the curved arrows here indicates the processes which emit heat. The emission of light from S1 to S0 state is called fluorescence. For the rest of my talk, I will focus on how fluorescence can be used as a tool to study structure, dynamics, and function in self-assembled systems. The figure in the left here is a part of my project during the internship at the University of Miami. In this work, we have studied assembly and disassembly of capsules that are formed by self-assembly at different pH using dyes. These dyes have polarity-dependent emission properties, which means that if the dye is in a polar environment, it gives a different fluorescent signal compared to when it is in a non-polar environment. What we observed is when the capsule is open, the dyes show a fluorescent signal at 490, and when it is closed, they give a fluorescent signal at 420 nanometers. And these can be differentiated on when you're collecting a spectrum on the spectrometer. Using fluorescence, we were able to show that in acidic environment, the capsule is open, and in the basic environment, the capsule remains closed. The figure on the right shows a hydrogel that is formed by self-assembly of two building blocks indicated by red and blue blue dots here. When these two building blocks form a gel, there are three possibilities. Either they can align by themselves like this, which is known as narcissistic self-sorting, or they can mix together in parallel fashion and they form something called social self-sorting, or else it could, and the way that they could organize can be random, which is called random co-assembly. When you see these gels on an electron microscope, which is normally used in the field of gels, you wouldn't really see the difference between if the gel has formed like this or like this or like this. So the three possibilities cannot be differentiated when you see it on an electron microscope. However, if these building blocks, which are indicated in red and blue here, are tagged with fluorescent dyes with different fluorescent properties, you can be able to see the different types of assembly that can be formed using fluorescence microscope. These examples show us how fluorescence can be used as a tool to study structure, both at the molecular level, like I showed you in the figure in the left, and also at the microscopic level, which is the figure in the right. I think by now I convinced you how fluorescence can be used as a tool to study structure. Let's now go into the dynamics part and see how fluorescence can help us to understand the motion, the movement of molecules inside the self-assembled systems. There is a technique called fluorescence recovery after photobleaching, which in short also known as FRAP. In this technique, we take a self-assembled system, we put the dyes inside the self-assembled system, and we study it under a laser scanning microscope. So what you do is you choose a selected area on the sample and you damage it using high intensity light. Once you damaged it, you no more see the fluorescence from that area. As you can see in this graph, when you bleach it, the intensity goes down to 0, which means that the dyes in that area were damaged by the high intensity light. Once this happens, the dyes that are outside that area, for example, the dyes that are in the green area or in this picture, will start to move into the white area, which is bleached. As a result, what you see is, as time progresses, you see the recovery of fluorescence in the circular area that was bleached initially, which you can see in the graph and how you see the recovery of intensity with time. This information will help us to know to determine the diffusion coefficients for the dyes. For example, how fast this recovery happens or how slow the recovery happens can give us information on how the molecules move in these assembled systems, and if we could be able to control these movements using certain conditions. The figure in the right is we have a self-assembled nanostructure, which is in the form of a micelle here, and the circular dots in red corresponds to the dye molecules. So the dyes which emit fluorescence, I told you earlier that the dyes give you this fluorescent signal. So they have different lifetimes. So once the molecule goes into the excited state, it can stay there for some time. I told you this is typically of the order of 10 to 100 nanoseconds earlier. So some dyes, depending on their structure, they glow for short time, but some dyes can glow for long time. For the dyes that glow for long time, they can travel between different regions in the nanostructure. Like as you see here in the figure, the dyes that are located in the free portion in this sample can move inside the micelle, and the dyes that are inside the micelle can start to move outside, and this movement happens during the lifetime of dyes. So these movements can be quantified using fluorescence by using a quencher molecule, which turns off the fluorescence for the dyes that are in its close proximity. In decoding function part, I discuss here two important goals of a drug delivery system. One, the drug delivery system should reach to the target site. Two, it should break and release the drug. That's whole what we expect from the drug delivery system. Figure in the left is a study done during my master's degree at Professor Singh's group in India. In this study, we show that the nanostructures containing dyes show fluorescence turn on when they experience an acidic environment. For example, at around pH 5 to 6, we see that we see a fluorescent signal coming from the nanostructure, as you can see in the figure here. Whereas at a neutral pH, the nanostructures are no longer fluorescent, and you can see them as dark if you see the sample on a microscope, you don't get any fluorescent signal. This fluorescence turn on and turn off mechanism based on the pH was used as a strategy for real-time monitoring of uptake of nanoparticles in cancer cells. Just to let you know, the cancer cells are in an acidic environment, which is typically of the order of 5 to 6, pH between 5 to 6. At this pH, the self-assembled structure that we made fluoresce. And when we get a fluorescent signal on the microscope, we can surely say that this self-assembled structure is now inside a cancer cell. Figure in the right shows a similar study where nanostructure loaded with drug enters into the cancer cell. As you can see in the figure, this is a nanostructure. It enters the cancer cell through these leaks on the vessel, on the blood vessel. Once it enters the cancer cell, it undergoes aggregation to build a bigger nanostructure. So what happens when this bigger nanostructure was formed is it cannot leave the cancer cell because the gaps are too small for this to go outside of the blood into the blood vessel. So which means that this aggregated nanoparticle can stay inside the cancer cell for longer times. Once it is in the cancer cell, the aggregated nanoparticle can undergo, can break and release the drug, as you can see in the figure here. Here's a timeline of the approval of nanomedicines in the clinic. Despite their huge potential, we only have about 15 nanomedicines in the market till now. And even there have been considerable efforts in the use of for the use of nanostructures in the current COVID-19 vaccine trials as well. One big challenge that's preventing this field moving forward, despite its huge potential, is not being able to control the characteristics of nanostructures that are formed. The characteristics, I mean, controlling their size, controlling their charge, and controlling when they can assemble and when they can break, and the conditions in which they can form as well. As we begin to uncover the information on the self-assembled systems through detailed structure and dynamic studies, I believe that achieving control on the characteristics of nanoparticles is totally an achievable task. In the end, I would like to thank my supervisors and mentors, without whom this journey wouldn't have been possible. I would also like to thank the members of the research groups I've been a part of during my scientific journey for their support. I would also like to thank sponsors who helped me to carry some of my work I showed you in this presentation. I also extend my thanks to SCWIST for the opportunity to be here today. I'm happy to answer your questions now. Thank you.
You could type in your question in the chat or just unmute yourself. Any burning questions? Yes, Tasha?
Well, very well done, Sree. That was a great talk. In your last, I think it was your conclusion slide, you were showing that most of the early sort of drug delivery, like the structures were made of, it looked like polymers or like some sort of polymer combinations. I'm wondering, is that, and then the other two structures, the newer ones sort of look different. I don't know, is it common for them to still be made of polymers, or what are they made of these days?
That's a good question, Tasha. Like we have like different types of self-assembled structures. Like you said, polymers are very prominent in this area, but we also have structures made from liposomes, the lipid nanoparticles, and even there are some structures where you can use iron nanoparticles, like which is again a different type of nanoparticles compared to the polymer areas. So these are still possible. The liposomes and the lipid nanoparticles, the polymer nanoparticles, these are like a mix of things that are happening here. The LNPs are, I mean the lipid nanoparticle, there've been very strong interest in that particular area these days. And if I like the COVID-19 vaccine, the companies which are using mRNA as mRNA as a drug for the COVID, they have been news about like using LNP nanoparticles, especially for the COVID related research as well. And I think like based on my experience at Precision Nanosystems, which is a biotech company at Vancouver, I really think that there is this, although like the approved part has polymers in a higher, there has been interest around that, but LNPs like are getting like huge potential has been observed around them these days. I hope that answered your question.
Yes, thank you.
Thanks, Sree. We have a question from Anna. Could you please again, how do you use FRAP as a tool to measure diffusion coefficient?
Okay, as a tool. Okay, so let me get back to that slide. The reason like how you can calculate the diffusion coefficient from this is like when you see the recovery of fluorescence happening after you bleached it, you can fit this curve actually to...
Sree, excuse me, we can see your slide, but it's stuck in the first slide.
Oh, okay. Oh, yeah. Okay. I'm sorry. I understood what's the problem. Can you see now?
Yes.
Okay, great. So what I mean, like how you can use FRAP for measuring the diffusion coefficient is like once you bleach the area and you see the recovery of fluorescence happening, as you can see in the figure with time, you can actually fit this equation, like fit this curve to mathematical models to extrapolate the diffusion coefficient for that particular, the molecules that are inside. So, I mean, this is like, it's not as easy as it sounds in this picture, but it like the fitting to a mathematical model will give you the value of diffusion coefficient. I'm pretty like, if you there are so many mathematical models that can fit to this equation. I'll be happy to share you some of the information later if you're interested in this part.
Thanks, Sree. Another question. Do you think this approach can be used to deliver drugs to treat conditions other than cancer? For example, it is hard to develop drugs that cross the blood-brain barrier. Do you think this approach has the potential to address this?
Yes, that's a good question. There has been use of these nanostructures for other applications other than cancer. The reason why I picked up cancer is because it's close to the area where I worked. I'm not really sure about the blood-brain barrier. I'm not really sure if I can give you an example, but I'm pretty sure that there has been research done in that arena. But it's not. I'm not really familiar with that side of the research, so I'm not. Yeah, I can tell you that there have been reports of the use of these nanoparticles for other areas of other diseases, but especially for the brain, I think it's outside my scope of my area, and I don't exactly know the information on that.
Okay. Any other questions? Feel free to unmute yourself or just type in the chat box.
I sort of had a follow-up actually to that question.
Go ahead.
So not necessarily in terms of the blood-brain barrier, but you were saying in tumor cells, because they're a lot leakier,
yeah,
the whole structure is actually able to get into the tumor cell or whatever and release the drug that's inside. Does that happen in other circumstances? Like, or is there other like healthier or not necessarily tumor cells where these full structures can make it through? Or is it that the drug is released before then and that's what actually makes it into the cell?
Okay, that's a good question. Like one of the problem with this area, using the nanostructures to deliver it to the cancer cells is they can even get accumulated in the liver, which is a healthy region, which is still healthy. It's not a tumor; it's still healthy. Yeah. So there are like a couple of reasons why these nanoparticles prefer to get accumulated into the liver, and I can tell you some stats that if you load like 100 out of the how much nanostructures you load into the patient, only about 0.7% actually reaches the tumor site. That's pretty low, but you still see some effect caused by those nanoparticles. The reason why it's pretty low is because most of them accumulate in the liver, and that's a very that's a key strategy. There are ways you can play around that, where you can avoid the accumulation of nanoparticles in the liver despite it being healthy. And I can give you the reason, some of the reasons that I studied on why there's the accumulation on the liver is preferable, is because one thing is yes, the nanoparticles can enter the liver cells. That's their size can still allow them to get into the liver cells. And also because when these nanoparticles cross the cancer cells, the blood vessel, they actually slow down. That's also, there is an interesting article around this entire the statement that I gave you, that the slowing down of nanoparticle when they encounter a cancer cell. Mm-hmm. This slowing down also was observed at the liver. So when it slows down, it has more chance to interact with the vessel, with the liver, with the blood vessel in the liver, and because it has more time, and also because liver cells can take up those nanoparticles, you still see an accumulation happening there.